That sounds good.
Great. This is on YouTube now. Okay. Good evening, everybody, to people listening from India. Am I audible? Yeah. So today we are delighted to host Professor Manu Prakash from Stanford to give today's webinar. So CCMB Biologue was a concept started by the PhD students of CCMB almost 2 years ago now, during the middle of COVID pandemic, because we couldn't host any speakers to give in-person talks anymore. So we decided to start approaching people to give webinars online, as was becoming the norm during those days. And then this venture has been quite successful. And so far in the 2 years, we have managed to host talks by almost 20 experts in life sciences across the globe in different fields of life sciences, such as biophysics and cell biology, even some science communicators, etc. And then how this works is that we usually ask the viewers who are both students and scientists at CCMB to suggest potential speakers they'd like to hear from, and then we approach them. And then through these channels, today we have the opportunity to have Professor Manu Prakash with us. And then I'd like to call Pradeep to give a short introduction to today's speaker, and then we can start the talk. Thank you.
Yeah, thank you, Vaidehi, for giving me this opportunity. So, Manu, that almost everybody knows, I think, in India. And like Manu needs no introduction, but still I have to do this job. So, Manu is actually faculty in the Department of Bioengineering at Stanford University. And he is being physicist by his training. And Manu's lab is majorly focused on two major aspects. And one of the aspects, Manu is trying to look biology through a lens made up of physics, basically asking biological questions with the context and insights from the physics. And the other major project that he calls like is a frugal science, basically where like he's trying to develop and deploy various tools and technology to make wider range of people to study or look at the biology at more clear eye, with more clear eye. And he grew up in India, that from Meerut to the IIT Kanpur. And his scientific career actually started at home, where he himself describes that first time when in his house, when he shifted in his rented house. So earlier it was occupied by a chemist, and then there was a lab in the basement where he and his brother, the first time they ventured into the science, they had a field and they played with the chemicals. And after that, during his school, when during his exam, but he mentions that he was trying like drawing the microscope drawing and labeling each and every part of it. But he has not seen any microscope. So that's where the quest to actually see and build the microscope started. And the first microscope that he describes, that he stole his brother's lenses and then he tried to make a microscope out of it. And later on, that quest actually ended up in with everybody, even myself also had this paper microscope, that is a foldscope, that now it's being reached to the millions of the people which they are trying to see the biology. And after that, there are various inventions are being done, like this paper fuse, where there is a centrifuge made out of the paper, where you can, where he has taken inspiration from the simple toys that we use during our childhoods, like the yo-yo and the spin with that. So like that, he had developed multiple things. Few of them are like Planktoscope, recently, if you want to study marine biology and various things that can be done. So from IIT Kanpur. After that, he moved to MIT to do his MS, and then he did his PhD in MIT. And then, if you see the various awards, like he has been the TED Fellow multiple times from 2009, 10, 11, and all those things. And he has been like awarded the Gates Foundation Global Health Exploration Grant, and then he He's MacArthur Fellow, and so like there's a long list of awards and long list of the patents. So the good thing that is that like he tries to bring the various tools to the common person he wants to do science. Manu, it's all yours.
So thank you so much, Pradeep, and everybody at CCMB for having me. I know it's 9 p.m. It's probably late, but we'll kind of keep this as a tradition of an after dinner talk. It's unfortunate that I'm doing this on Zoom rather than actually being there on person, but maybe someday that will happen. I'm going to share my screen. And I think before I start, one of the things that I just want to mention is I'll talk about some of the very basic science, very fundamental science oriented questions that we work in the lab. But I think as Pradeep described, what gives me great joy is the same sets of discoveries that primarily arise from just observing our natural world. We deeply care about sharing the tools for those so that every single person, not just the context of somebody who's a professional scientist, but anybody who you interact with, anybody you talk to in the community, they have opportunities and chances of deeply understanding how absolutely remarkable this natural world is. And so I think, my hope is maybe I'll primarily focus on kind of this basic science questions, but at the very end, it would actually be fun to talk briefly about what can we do about making sure that not only tools are accessible, but mentors are accessible to people who want to venture into biology as their next adventure. Okay, but for today's talk, I picked up something that I've been thinking about for a while, this will be presented in a context of puzzles. There are two stories that I was thinking of sharing. One of them is on bioRxiv, another one that we're about to post on bioRxiv. But it's an idea that we've been working on for a while, which is the role of geometry in cell biology, and very specifically in the context of topology. And I'll describe a little bit of what I mean along the way. It'll be set up as a few puzzles. So, you can entertain me, think a little bit about these sets of puzzles as we go along. And then before I start, I just want to say I have a wonderful opportunity to work with just a remarkable group of students, postdocs in my lab. Many of them are there. Many of them have graduated and moved on to really wonderful careers. This is really one of the greatest joy that we all have, to engage with individuals over a period of time. And I just find it both remarkable, and also the fact that we have that privilege. And also I get to talk about many of these sets of ideas with the work that we have done together. I'll talk about the individuals involved in specific projects when I'm talking about the projects themselves. As a lab, we focus a lot on physical questions in biology, and the other contexts of making scientific tools accessible. And many times the process is quite circular. In the access of making scientific tools, we also end up finding completely new novel biology. And actually, the two projects that I'm talking about, it did end up that way, where venturing in the act of making these tools more accessible, we actually discovered new biological puzzles. So let's just take a step back. And what I want you to think a little bit about in just the first framework for this talk, but in general, is this idea of recreational mathematics. Recreational mathematics has been around forever, since mankind started thinking and sharing stories. And I don't know how many of you know Martin Gardner, a phenomenal writer that really advertised and popularized recreational mathematics. And the principle of recreational mathematics is questions and ideas that you can share and ask in a single sentence or two sentences that literally anybody can understand the depth of the question. it should be simple to understand, and it might be at least the question, and it might take the most advanced fields of mathematics to really be able to decipher and figure out how to tackle a problem. We will talk about topology. Topology has a lot of these types of puzzles associated with it. If some of you haven't had the chance to read Martin's work, I would highly, highly encourage it. It's just so inspirational. Um, I've been thinking about this idea for a while, and what I've been framing for my own self, and I'm now wondering whether people listening and the people hearing here, how you would think about it, is this notion of recreational biology. What if we forget for a second, all the important things, of course, from a biotechnology perspective, for biomedicine perspective, why we work on certain questions, and purely ask them just to be able to satisfy our curiosity. But the other premise I want to apply to recreational biology is that the question has to be explained in a single sentence or two that anybody can understand. I'm going to choose two examples of those types of questions. And I think you will find in your own fields, it will rephrase the sets of questions, but that leads to new insights. And especially the framing that I'm excited about using in this is the framing of paradoxes. On the right is the picture of Max Delbruck, slightly late in his career when he had done much of what we know him for. And he was thinking a lot about his motivations. He was challenging and asking completely new questions. But I love this sentence that he phrased, which is, he was trying to describe his motivation, where he said, we'll run into a paradoxical situation analogous to that to which classical physics ran in its attempt to analyze atomic phenomena. And what he's talking about is much of atomic physics and much of our ideas of the small scale in physics. It was very hard to visualize, very hard to do the kinds of experiments. And so it was primarily built around paradoxes, that if you're going to go with a certain set of an explanation, you end up at a paradox. And in the act of solving that paradox or that puzzle, you actually decipher something remarkable about our world. And he says, this, of course, has been my ulterior motive in biology from the very beginning. And I often think about the biodiversity of life on our planet by itself as filled with these wonderful puzzles and paradoxes. And we will take a few examples of those today. So on topology, let's just take a quick break. I want you to think about food. This might be a food that some of you like. Here is a donut. Of course, I'm bringing up the donut for a second as a topological object. As many of you know, a donut has a hole in it. And no matter how much you deform it, you cannot actually turn this into a sphere. Just fundamentally, a sphere and a donut have fundamentally two different properties. And then just to give some homage to Martin Gardner, if some of you get bored in this talk, you can use pencil and paper and figure out by the end of this talk, how many pieces can you cut a donut with 3 intersecting planes? So you will get a sense for how topology is remarkable. And of course, we will come back to this idea of cutting donuts. There is actually a reason I'm mentioning this. So 3 straight planes, how many pieces can you cut a donut? Okay, so why am I talking about topology? First of all, from a mathematical perspective, you can think of a sphere and a brick are topologically exactly the same, while a donut and a cup are exactly the same, while a scissor and a kettle, for example, is topologically exactly the same. And this is called handle in a mathematical context, in which you can see that deformations of a cup can transition it into a donut, but no amount of deformations will lead a donut to turn into a sphere. Now, this is conceptually a very powerful idea in, of course, mathematics, but also in context of physics. There is, I don't know if many of you have been on conveyor belts, for example, and the original patent for conveyor belts are actually Mobius strips from a perspective of how wear and tear in these strips works. You want all sides of the strip to wear at an equal rate. There are ideas of topology in fundamental material science, topological insulators, topologically preserved properties. So we really look for, in terms of thinking about the intersection of geometry and physics in a lot of problems. And the thing that's valuable in respect from a biological perspective is you could ask, oh, where does this context arise in biological problems? Many of you who might be studying a developmental context might realize that our entire prescription of growth and development in biology in terms of embryos is driven by a topological context. We are all, in some sense, toroids and donuts with two openings. You can think about, where does that even come from? Are there organisms out there that only have one opening? You can think an example of an hydra that has topologically a different object. Also, of course, people have thought quite a lot about topological challenges in DNA itself, just unwinding of DNA. There's a lot of work in terms of tissues. People are starting to now think about when you're developing tissues, and especially organoids, how do you generate the kinds of cavities? What would those structures actually look like? And again, this concept has really been growing in the field of active matter as well. But I'm going to focus on a cellular context, and a question that has been a quest for me for almost the last 15 years. I've been thinking about cell biology. Is a very simple question. Do toroidal cells exist? Why is it in nature that we see them or we don't see them? This is something that I started looking and searching for a long while, and it took almost this long a time. And I'm actually very happy to share that we have actually made some really fun progress on this. So I just want you to think about a donut cell. Could that exist? Funnily enough, E.M. Purcell talked about a donut swimmer, which was not in a biological context, but they have profound implications. Membranes, biophysicists have made donuts for the long time, but ironically, we don't find that many donuts so easily in the context of cellular perspectives. And one of the fun things about this is there have been some remarkable experiments in cytokinesis where Ray Rappert, who many of you know from the cytoskeleton world, actually came up with a brilliant idea of taking a sea urchin egg and squeezing it with a glass needle to give it a topology of a donut, and asked a very simple question of how would that donut divide. And what he remarkably discovered, and I think this is a great example of recreational biology, he asked a really simple curiosity, but what he realized is, if you were a donut and if you divide from a cytokinetic ring perspective, although you have a single division ring, the cell has not topologically divided into two separate entities. Because if you cut a donut with a ring in one place, you see the donut is still connected. It's open now. It's not a donut anymore. It's actually equivalent to a sphere. But you don't have two cells. You still have one cell. And I'm not going to talk about this, but you should take a look at and see what he discovered, which is literally the communication between the cytokinetic ring and the spindle determines where these rings would grow. And it actually turns out cells are clever enough that they get out of this puzzle. And at the second division, they actually make multiple rings to be able to then, after two divisions, we still get 4 cells, which is actually quite puzzling to think about. Okay, so I think, I often go back to nature and looking at the context of thinking about the breadth of biology that exists. So my premise was, out there somewhere, there's got to be these types of cells. Let's look for them. First of all, the geometry I'm talking about is just one example of a geometry. We are really fundamentally also interested in just what cell geometry does. I think it's underappreciated if you just look at all the taxa, just even in eukaryotic protists itself. Geometry plays an incredible role in the context of defining the class of behaviors and the class of environments that are possible. As a lab, we study quite a lot of work in thinking about how organisms encode behavior. And what we are discovering over the last 7-8 years is much of that behavior unlike in a higher organism, like a system that has a neuronal system, much of that behavior is actually really encoded in the geometry of the organism itself. So this has become really a quest. And while thinking about this, we made an important leap in the work that we do as a lab. Since CCMB, you have 3 communities of ecologists and molecular biologists and technologists. This is a talk that will be at the intersection of these 3 things. I will talk about ecology, I'll talk about cells, but I will also talk about a new technology. But that is driven by a very simple question, which is we should do cell biology in the context of an ecological framework. How do I even explain all of this diversity if I just don't even know where an organism lives? And this is, I love this example I heard from a friend of mine who was giving a talk about the Drewer's rhino. This happens to be on the left, a painting from 1515. It's a very famous painting. And the ironic part of this painting is it was actually made by a Portuguese painter who had never seen a rhinoceros. And the only way he knew about a rhinoceros is stories from India, where travelers who had described seeing a rhinoceros then described it to another traveler that described it to another traveler, and the story reached Portugal. And then in the end, he made this famous painting for the king. Ironically, he got a lot of things right. Rhinos have horns, they have some kind of a thick skin, but they don't have armor. And I sometimes feel cell biology is stuck in a Drewer's rhino. Unless you really deeply care about, you might be studying macrophages, you might be studying neural cells. Unless we truly understand the environment that they respond in and the environment that they evolve in, it's very difficult to really understand their behavior and what they are trying to do and achieve. This is almost like meeting a rhino in a petting zoo or meeting a rhino in a savanna in Africa. You are dealing with completely 2 different animals. And I think this has been a recognition in cell biology community, the fact that we pretended a lot of our many principles early on came from cell cultures that grow in environments that don't actually mean anything, or cells that have been separated from their context and just only grown in culture for the longest period of time. So as a lab, we have focused significantly on spending time out in the wild, bringing our scientific tools and capability to the field. And I just want to share with you how much fun that is. And I encourage anybody and everybody to take their science outside their lab. It enriches you in ways and forms that I cannot even define in words. This is what it's meant. For the last 3 years, we've spent a significant time on research vessels. And the discovery that I'm about to tell you was actually made on one of those research vessels up on the top. That's Kilo Moana. This is a picture taken off the coast of Hawaii. We were lucky enough to get a spot. While we took all our instruments, on the left is, I think, RV Sequoia, all the way up in the Arctic. And this is a trip that 2 students of mine, Rahul and Ian, just finished all the way in a trip from Seattle to Hawaii back to Alaska. All the way at the bottom is a vessel that goes out down to Antarctica and Palmer. Another student in the lab just came back. On the right is another small vessel that we've used. These vessels are very hard to get access to, so we also just use fairly small vessels that we can find. I'm actually quite excited about the fact that there is a lot of momentum now on setting up marine stations across the coastal lines in India. And I just want to share with you what it means sometimes to do this type of work. And on the right, this is a video from Ellie of the Drake Passage, and you can see the waves. And so this is not for the faint-hearted sometimes. It does get quite rough. So it's not all fun and games. But I want to make sure, I think the joy of what you get in the end is actually quite remarkable. Okay, so I think I'm going to jump to tell you the first discovery of the story that I'm excited about. We also get a chance to actually work at this wonderful place, which is the Hopkins Marine Station, which is our local backyard, which is where Stanford has had the marine station. I had the luck to essentially have the lab there now for the first time. We have some lab space there, but it allows us to really test many of our ideas in field settings. So let me tell you about the first story of this, a toroidal topologically complex cell. And we're going to talk about this in the context of the ocean. And this work is done with two postdocs in my lab, Adam Larson, who has a biochemistry background, and Rahul Chajwa, who actually comes from India and really transitioned from working in purely physics-based problems in sedimentation and joined my lab recently. It's been really wonderful to work with both of them on this challenge, and much of the work that I'll describe was done by both of them in the lab. This work is also enabled by a tool that we have developed, which we call Gravity Machine, which has been a large project in my lab on how to really do microscopy in the context of the ocean. So I'll briefly describe that, but Gravity Machine has been, was part of Deepak Krishnamurthy's and Hong Chuan Lee's PhD, but many members in the lab, including Ellie, Ian, Pranav, Ethan, and several other members have been helping develop this tool, and we're trying to scale this tool slowly. So the question that we're interested in is trying to first understand life in the ocean, because the organism that I chose for this particular thread lives in the ocean. And the image that I want you to have is, think of you as a scuba diver and imagine, very much like, you know, mountains, you can cut chasm. Imagine if you could cut a chasm in the ocean, what would you actually see? And we are really interested in how life distributes itself on the vertical axis. How would that actually look like? You know, in the World War, a famous discovery was actually made, which is life, and especially I'm talking about planktonic life forms, are not stable. They're not stationary along the vertical axis. And as the sun moves, so you're seeing this in this animation, as day and night passes, living organisms, including single cells, would travel hundreds of meters to even a kilometer to come up and come down. And if you do a little calculation, you can actually show that the amount of energy that these organisms spend per day is actually equivalent to the entire energy budget of United States. This is the world's largest biomass migration happening on our planet. You know, much of the biosphere on our planet is in the ocean, and you can really think about what are the decisions that every one of them is making at any given point of time to do this massive migration. If you were a diver, this is what you would see. This is right in the middle of this migration, billions and billions of plankton, single cells going about their business, doing this marathon every single day. The reason we chose to study this particular problem is also has a societal context to it, which is much of the CO2 that we have all been pumping for the last 100 years into the atmosphere is actually absorbed by the ocean. It's absorbed by the carbon that essentially is dissolved in the ocean, that is then converted into biomass by biological processes into the mass of these organisms. And when these organisms die, it essentially sequesters carbon, which is called a biological pump. Roughly around 20 billion metric tons of CO2 is removed every year. And if this pump was to stop, we would go from 400 parts per million to 600 parts per million overnight. So it's a very important biological process. And unfortunately, in many of our mathematical models associated with this, biology is actually incorporated in a very simplified manner because nobody has actually watched these organisms make these transitions. We don't really understand what types of complex processes are at play. So this was really the big motivation, because we would like to really be able to build these kinds of, from cell biology perspective, go all the way to environmental models. These are some of the best examples of current existing environmental models. This is work from Mick Fowler's lab at the MIT Darwin Project, where you're seeing a simulation of life in the ocean. Ironically, this is done in the context of there are mathematical frameworks that are defining what the cells are doing. So this color represents different types of plankton. This is purely in simulation. It's in silico. Of course, with satellites, we can measure some of the chlorophyll so you could back check it. But, you know, in the end, a cell is a very complex complex object and it's making its decisions across its life cycle. And this life cycle question will become important. So here are just a few motivations to really think about why we care about even understanding what does a cell do in an open ocean. And you might wonder, Manu, you talked about the environment, but you chose the ocean, which seems like a massive infinite bath of water. Where is the ecology? Where is the complexity of environment? And I think here comes the biggest surprise, that ocean is an incredible system to ask these class of questions, because everything in the ocean can be understood in the context of vertical stratification. What I mean by stratification is an idea that, very simply put, if you shine light in water, light gets absorbed. So the deeper you go, the darker the ocean gets. And around 150, 200 meters, there's absolutely no light. It starts to get really dark. And after 1,000 meters, it's just abyss as an ocean. So the ocean is divided into the euphotic zone, where there is enough light for photosynthesis to be viable. Then you are in this mesopelagic region where photosynthesis by itself would not be viable. And then, of course, you have the dark deep sea where there's absolutely no light. Not only light, pressure. If any of you dive or even snorkel or even swim, the deeper you go in the water, you actually feel the pressure. Every 10 meters is 1 atmosphere. So for 100 meters, we're talking about 10 atmospheres of pressure, temperature, salinity. Any aspects of these types of physiological parameters are actually varying along the z-axis. We're going to use the term z. So just as an orientation, if you hold your hand, xy will be in the plane of the ocean, and then we'll use the z-axis as depth. And an important aspect of this from a cell biologist perspective is, life evolved in the ocean. And you can ask yourself a question for a cell: is a cell heavier than seawater or lighter than seawater? And it's actually a really fascinating question. If you think about cytoplasmic density, you can measure it in the lab. Often it turns out for majority of the cells, it's 5 to 10% heavier than seawater. And this will become relevant on average. Because if you drop a coin, for example, if you had a coin and you dropped it in the ocean, it would sink because coin's density is heavier. And there is a famous Stokes Drag term. If we just walk through this equation, you can see U is the velocity of sinking. You can see rho is the density. And I'm subtracting the cell density from the water density because it's the relative density that matters. And importantly, it depends on the size of the cell, which is the product that G is the gravity constant and R square. And then, of course, you have mu, which is the viscosity. So the viscosity also changes a little bit, but in this case, it's roughly constant. But you have a product of size of the cell and the density of the cell that determines how fast you will sink. But if you were to sink, you can imagine you reach an environment which might not be viable for photosynthesis. So we're going to ask this puzzle is, how do cells manage this kind of a trade-off between where you want to be in the ocean? How do they make these sets of decisions? And ironically, we will ask this question in its simplest form for cells that for the last 200 years, we have called them non-motile. We have often thought that if a cell does not have a flagella, for example, or any cilia, any kind of an organelle that's dedicated to motility and it's living in an open aquatic water, we assume that it's non-motile. And I'm going to tell you that actually is not true. So we're going to ask this in the context of non-motile systems. And ironically, there is actually a trick around this. So let me take you to 2019. This is the trip that Adam, Deepak, Hangchuan, and I took. And we had built a tool, and we discovered this very exciting discovery that we were casting our bottles. What you're seeing in the middle there is called a CTD. It's a gadget that I can drop into the ocean and open these bottles at any known depth. And at 75 meters, we open the bottles. And out came this incredible cell that you can see on the right. It's roughly around 200, 300 microns. It's called Pyrocystis noctiluca. And one of the things that is remarkable about it is we saw it at depths that we didn't expect it to see, because 75 meters was low enough for this photosynthetic organism. And we started wondering what is going on. Just to give you a sense of where it is a dinoflagellate. So if many of you know ciliates, in the tree of life, you can see right next to these ciliates in the alveolates are dinoflagellates, dino implying two, and flagellates are two flagella. Ironically, Pyrocystis doesn't really have in this cell stage any organelle which is associated with swimming. Many of you might have heard of dinoflagellates, and especially Pyrocystis, in the context of bioluminescence. I don't think we still really understand why bioluminescence works and what is the purpose of bioluminescence, but many of you have seen photographs to the right where this mystical, magical light is produced. That's actually produced by these single organelles. So it's actually quite fun. Although I'm not going to talk about bioluminescence, all the cultures in our lab glow. So from time to time, we like to just go to the lab and just shake them at night. So we tried to look in to see what has been done in the past in exploring the distribution of this cell. And we found this really remarkable paper from Swift in 1976 that this specific dinoflagellate is actually a deep diving cell. It has the capacity to dive all the way to 200 meters. And because it's photosynthetic, you have to have this cell have a capability to come up, because if your progeny doesn't come up and you're photosynthetic, you can see how this goes. Evolution works this way, that if every cell cycle you keep sinking, at some point of time, you will cross this light barrier, and then it will be feasible for you to actually stay alive. And this is actually also true for a couple other species, but we're really primarily focusing on this specific cell. So here's the challenge. How do I study a cell that's traveling 200 meters in a lab setting where I want to do cell biology? So I want to really see what might be going on in this cell as it's making decisions. So from a microscopic point of view, I care about microns and hundreds of microns. I want submicron resolution to see this process. But I also want to recapitulate a travel of at least 200 meters, and in my mind, almost kilometers. And one simple way of thinking about this, hey, let's build the world's tallest microscope. And we actually did start thinking about it. We built a 3-meter microscope. And then we realized it's kind of a tough ordeal. And we came up with a trick that we call the gravity machine. So the problem here is that these transitions are happening over long time scales, and the length scales involved are also 6 to 10 orders of magnitude. So you're left with this really challenging puzzle. And the problem is, how do I even make a 100-meter microscope? And any of you who have built anything that's even a couple of meters, it's not stable. Making a microscope at that scale seems hard, until we came up with this idea of what we call a gravity machine, which was published a couple of years ago. It's accessible. We call it scale-free vertical tracking microscopy. And the simple way to think about it, it's essentially a treadmill for single cells. So many of you, if you keep pets, you can put them on a wheel, and although the pet is running, they're not actually going anywhere in the lab frame of reference. We invented a similar trick. You should think of it as a virtual reality glasses for single cells, where you are looking at a microscope that you see on the right, which is tilted 90 degrees to the traditional way. Every time we make microscopes, why do we have them aligned to the gravity axis? Here, the microscope optical axis is horizontal. And instead of putting biology in like a confined tiny cover slip, where of course you're not seeing the original behavior that you're bound to see, the cell or an organism is in a loop. And you can see why there is a loop, because if vertically there is an infinite distance that you can travel. So if the cell swims up, we actually spin the wheel down. If the cell swims down, we spin the wheel up. And so in the lab frame of reference, the cell doesn't go anywhere, but in its own frame of reference, it's actually climbing infinite distances along the vertical axis. So we had this tool, and we took this powerful tool, actually 2 kilo Moana, and started looking at what this cell is doing. And you're actually going to see the first time we observe this organism, there's a copepod in the back. This is a video from the field on a deck of a ship. This is what it looks like working in the field. We have just collected this cell from around 75 meters. We run back. It takes us around a couple of minutes. We put that in the wheel. Ironically, I love this video because we were right in the cusp of a storm that was about to come. And so you can see the storm is actually maybe around 2 hours away. And unfortunately, right after I took this video, we had to unpack everything. The ship had to turn around, and we got in trouble. But we had gotten a lot of good data for a couple of days before we had to turn around our mission. And the type of data that we collected is the same cell rising and sinking in different cell cycle contexts. So what you're looking on the right is a cell that's rising, and you don't see it rising because you're swimming with it. You are in its own frame of reference and its vegetative state, while the dividing cell is actually sinking. There is no motility, there's no flagella, there's nothing attached to it. How the hell is the cell doing? And of course, some of you have already started thinking about it. The cell density is changing as a function of time. Now, what I love is if you look on the left here, Adam calls this the whale bar. The kind of migration that we are interested in is, if roughly a whale is around 30 meters, this is multiple whales of a migration. Whales make these sets of migrations, but now here is a single cell making this migration. And the first observation that we made is that cell cycle was coupled to this migration. It's actually quite interesting to think about that a cell will choose to live in two different environments. And actually, we know this, and since I'm running out of time, I'm going to just tell you this, that in its vegetative state, the cell needs light and it's trying to photosynthesize, it's making a lot of proteins, it's biosynthesizing, but also it actually needs nutrients, and some of the nutrients, specifically nitrogen, in much part of the ocean are found deeper. So the cell does need to go deep to get these types of nutrients that are not available in the mixed layers. So it's a paradox, but you don't want to go too deep, because if you go too deep and you can't come back up, your progeny is going to get lost. And that's literally evolution. So how do you balance this tricky fate? The second surprise we made is we brought this cell to the lab, and then we started looking at it and also at the field itself. We saw a remarkable event that happens in this cell, which is during cell division, it massively inflates like a balloon. So what I'm showing you here is literally 6-fold volume expansion of a cell in less than 10 minutes, which, for any of the biochemists in the room should be puzzles, because if I took any enzyme biochemistry and I told you to dilute it by 6-fold in volume, you're going to be like, oh, none of the kinetics is going to actually work out. How the hell can a cell, and of course, in 6-fold, how could you even make that much amount of material for the cell to grow massively in size? 6 folds in volume. I'm going to just play that one more time. And again, this also happens in the context of sedimentation. But there is a trick here, which the trick is that while this inflation was happening, we also saw that the cell was starting to turn around. That as this inflation is happening, instead of sinking faster and faster, it slows down. So this is the key clue. So first of all, we wanted to know, what is the cell cycle? We essentially imaged the cell for 7 days in day-night life cycle. So this is a video that Adam took. It's a remarkable video. I absolutely love the colors here. And the first thing you see are the plastids going in and out. You see them being deployed, and in the night they get taken back in. And that's a process that's been known for many organisms. But notice the division. The moment the division happens, this is a 7-day video. Any cell you watch when it's about to divide, there is a theca right outside, which is the cell wall, and the cell wall will enzymatically break right there. It disappears, and then boom, it suddenly inflates. So the question we wanted to really understand is, what on earth is going on? Why would you build such a crazy division cycle? And one of the contexts of this is, how is this even possible? This should not be physiologically possible for a cell to do, for it to be viable for all the living processes. And here is where topology comes to the rescue. Ironically, when you look for something, you never find it. So I spent an infinite amount of time poking around in ponds and things, looking for a topological cell. And when I'm not looking for it, we were handed a cell that has an incredible topology. So now I'm going to show you light sheet data, which is volumetric data for this cell. And you're going to notice its bizarre topology, which is the cell is not just like a traditional bag. The cell is actually a cytoplasmic reticulated network where you can see these strands going back and forth. They're all connected with each other in this very complex network. But at that same time, they are, in this mathematical jargon, it's called an N-torus. Like, it's not just donut, which also only has one hole. It has literally hundreds of holes. and it's topologically connected. But for the first time, it made sense, because now if you inflate this object, what's actually happening, and this topological object is entangled with a vacuole. And I don't know if it's obvious here, but you notice that some of these strands go through the cell, while many of them go through periphery. That's what I mean by entangled. It's entangled with a vacuole. And we can prove that by doing TEM, and we actually find that these strands of cytoplasm don't actually change volume. Adam went on to measure the volume, a lot of work that went in, and we showed that actually the vacuole is 2 orders of magnitude larger in volume. And and many of you who might think of plants, you might be thinking about plant vacuoles have a certain analogy to this, which has this tonoplast. But then the topologically, the cell is entangled with it, so you cannot separate it. But when the cell expands, what's really happening is the vacuole is growing like a balloon. And what we started thinking about now, this is just that light sheet data of that ballooning process. And you can see that this meshwork is being stretched massively, but not inflating. And we can quantify this in the context of volume. So what I want you to remember as a picture, think of an incredibly complex donut. On the left, we talked about a donut in the very beginning. Now you can have 2 holes, you can have 5 holes, 10 holes, you can have hundreds of holes. And that is the topology of this cell. What I love is Adam actually went on to print this object, and it's just absolutely beautiful. I have it. I'm running out of time. Otherwise, I would have actually shown it to you. It's quite valuable to embody your data sometimes. Here is the challenge. The moment we saw that expansion, we started thinking about its molecular mechanism. And I made this kind of a remark that, oh, let's look for aquaporins. And then essentially we did a transcriptome for the cell, and we actually found 4 or 5 candidates for aquaporins. Many of you know aquaporins from fundamental for biology. Almost every cell needs to maintain the amount of water it intakes. These are some AlphaFold predictions of these aquaporins we have identified. There is the closest one is AQP1. There's also a class of regulatory contexts. I don't have time to talk about it now, but many of the calcium-mediated kinases. The other thing that we are actually very interested in is what might be regulated these aquaporins. But we did classical experiments to show that, first of all, the rupture of the cell wall is essential for the cells to inflate. That's happening enzymatically, but we can do that mechanically. When the cell is about to divide, we go in and poke with a needle and we rupture this dinosporin. The moment the water, which has a lot of calcium, touches the cell, boom, you're going to then see an expansion event. So that's the signal, which is as the cell wall dissolves. And of course, the reason I brought up aquaporins, the only way you can rise and change your density if you could bring in pure water into the vacuole. So remember, I said seawater is denser than water. But with aquaporins, if you could bring fresh water into the vacuole, it's almost like a submarine, a ballast. By bringing fresh water that has low density, you can now reverse and you can actually start rising. So again, as you see, why this is so important to think about in the context of cell cycle. And these are examples where we can remove calcium from the seawater and we can actually stall this expansion. So we know it's driven by calcium. Okay, so I want to get to the mathematical context of this now. So I described the cell cycle. You get a sense of there is a vegetative phase, there is a very fast division phase. Now we want to understand the cell cycle in the context of ecology. And mathematically, one of the things we started writing, and this is work with Rahul, we started writing cell cycle just as a cell phase, in the context of activity. You can think about a clock. The cell goes through a clock. And the reason this becomes very important to think about is fundamentally the definition of cell cycle. When we say a daughter cell, and eventually after set of growth, the daughter cell has to become exactly what a mother cell was. So all the parameters that we care about in cell biology, eventually in the context of cell cycle, have to periodically come back for that mother cell to then give rise to the next cell. So this is why this set becomes mathematically really elegant to actually model. And so I think we're going to divide this into this delta phase, which is this inflation phase, and the vegetative phase, which is kind of the growth phase and the pre-division phase. Now, the thing that's marked with the red arrow is the delta phase is only 8 to 10 minutes, while the blue arrow is actually 7 days. So I want you to remember that this is not to scale. And just as a reminder, we already discussed this, the velocity is dependent on density, but now density will be dependent on cell cycle, and radius will be dependent on cell cycle. And of course, the ecology is a function of the depth, and that depth is in seawater is also dependent, the density is dependent on depth itself. So one of the things that falls out into this is we're going to break this into models. And I think since I'm running out of time, I'm just going to show you the final equation that comes out. But the important thing here is to think about cells are in their cell cycle changing their density. In the rapid ballooning phase, the density falls dramatically for it to turn around. And in the long growth phase, the density rises up slowly. So what you're seeing, the x-axis is in a log time scale, where you're seeing rapid decrease and a long rise. And these two, the balance of these two will give rise to a bouncy up and down motion for hundreds of meters. And of course, one of the mathematical tricks we end up using here, which was a brilliant idea from Rahul, is we ended up using this context of boundary matching, where we can now construct this in the context of two limits, and then we map the asymptotes together that give rise to a density term here. So now all the red dots in this plot are actually measured experimentally, and then the black curve is this mathematical prediction. And on the left axis, or the x-axis, you can see is essentially the cell cycle. So we have measured certain portions of it and then mathematically constructed, because there are certain regions that we cannot as yet map and measure. 7 days is too long a time to track a single cell while it's sedimenting. But because there is periodicity in the definition of cell cycle, we can actually map these two densities. So one of the fun things about this is we can write this now into a complete single model. And the reason I'm dwelling on it, you will see something quite beautiful falls out of it. I described the Stokes equation, which gives us the sedimentation speed, which is a function of density and radius. I described the ecology because the salt water changes as a function of depth. So now you know that depth is incorporated in that equation. Then finally, I define all the parameters as a function of cell cycle. And then we non-dimensionalize these terms, and eventually, a little bit of a math later, this beautiful equation falls out. Now, I absolutely love this because in a single equation, we have captured ecology, which is this beta z term, which is the density. We have captured cell growth and cell cycle, which is a function associated with the density. And we have captured behavior, which is the red term of where does the cell want to be. And so it's a combination of ecology, physiology, and behavior all showing up in the same equation. And because this is analytical, we can actually ask what would be the fundamental cytoplasmic density beyond which a cell cannot rise enough. We call this idea gravity trap, which is life must have, and again, for the last 4 billion years of life of this planet, gravity has never turned off. Lots of ecological parameters have changed, but gravity has been fundamental to evolution of life on the planet, and especially a lot of life evolved in the ocean. You end up finding up these very critical parameters, and the best way to see this is right here in the plot to the left. You're watching trajectories of two cells that we are initiating at two different densities. On the x-axis on the right, this is a phase space of cytoplasmic density. One cell has the capacity to do this up and down, and we call it that it can levitate in the ecological conditions, while the other cell that crosses that cytoplasmic density, no matter how much aquaporin, no matter how much ballooning you do, you will eventually sink. And ironically, this critical density of the cytoplasmic density, as you see, that's rising almost vertically, is exactly where most of the cytoplasmic density that we find in life in the ocean. So it's, I mean, you know, it might be a coincidence, but we find it remarkable that such a simple mathematical framework actually might capture a fundamental parameter if you were to think about from a biochemical perspective, why is it that a certain cell has a certain kind of a density? Of course, in terrestrial life, other kinds of challenges came about because gravity is much higher when you are outside the water, because the net density effect is mediated a little bit by the fact that you're in the water. But when life evolved, this must have been a fundamental parameter at play in terms of thinking about if you're photosynthetic and you really care about remaining in that euphotic zone. And you can think about this because eventually the depth of the ocean is such that only the life that could survive in those light zones would actually be able to survive. Okay, so in summary, what have I told you? I've told you about the longest cell migration of a non-motile cell today, 100 to 200 meters. We talked a little bit about this crazy inflation as a way to measure and change your position in a water column. And ironically, it is only possible because of cell topology. And now we are very excited because I'm now searching for many other cells that might use and exploit this topological principle. But it's a fundamental mathematical principle that you cannot avoid if you're going to do such a large inflation event so quickly. And I think what I'm very excited about is a very simple framework that allows us to combine ecology and physiology to now map these ideas to many, many cells in the ocean. I'm about to stop, but I think one thing that I want to mention is if some of you are interested in reading about this machine that we have built, you can go to this website called gravitymachine.org. We actually work on a large number of organisms in the ocean. Some of you have seen our work in starfish larvae, for example. So there is a very broad range of work that we do in plankton biology. We work on diatoms, we work on coccolithophores and viruses. And we are starting to create and share these databases associated with these behavioral data sets for a large number of cells. And now, of course, I only told you about cells that don't have flagella, but we also actually study swimming organisms, map dial migrations with these. And just to give you a context of what single cells do as a behavior, this is a flagellar dinoflagellate that does use its flagella, and it's actively swimming and sinking. And we are really trying to understand how do we account for this diversity that exists and the ecology that exists, to really be able to see how and why, what are the constraints around life living in this gravity axis. I'm going to stop here and transition this into questions, but I am tempted to just show you all one picture associated. There was another story I was going to tell you, but I don't think I have time now to talk about it. But the one picture I did want to show you, which might be right here. I want to end with this picture because, again, remember I mentioned this notion of sharing science. I wanted to share this notion of how you make discoveries, how by being out there, you can discover new objects to think about. But I think that comes with a responsibility. And that responsibility on every one of our shoulders is to share not just the joy of science and the discoveries that we make, but share the capacity to make those discoveries to individuals around the world. And as phenomenal biologists, all of you listening in this room, you have that capacity to share the tools of discovery with just communities around you. And I think it's been, it's the 75th birthday of India. So I'm actually very proud of one of the programs that we ran with DST that has led to full scope distribution across India. And so when I meet a person, I see a biologist in all of us. And I think if you take this to heart and you think about how deeply the principles of biology actually govern and rule our life, the air that you breathe, the food that you eat, the communities that we live in, and who we actually are, and the fact that we have locked up this capacity to explore the natural world only for the few of us who are privileged enough to have now pay for our curiosity, it's just not right. And so I think my hope really is that if you take something away from this talk, remember the joy of exploration comes with a responsibility of sharing the capacity of explorations with an everyday person. So I hope you take that, and I'm happy to take any questions. I really apologize. This ran way longer. I wish I had more time, and I would have gone through much more slowly, but I'll open up for any questions.
Yeah, Manu, it was really an amazing talk. Like as usual, like to hear from your videos. Yeah. So I'll go through this couple of questions that we have received from the audience. So one of the questions that we got from the Niranjan, what he's trying to say that we move with the help of legs and bacteria with the help of flagella, and the chromosome by spindle fibers during the cell division, and ions by diffusion or osmosis. Then how does the mRNA gets transported from nucleus to cytoplasm? Not only mRNA, even proteins and other biomolecules get transported. How signal peptide sends the destination? What are the driving force behind these biomolecules?
Yeah, I think a beautiful question, Niranjan. And again, I can see in your thinking, you are thinking about the physical context of this. I often think about this, that this is also valuable and important to do back of the envelope calculations. I don't work on mRNA transport. There are probably much better people than me, even in this audience, who could talk about this. But one challenge that I would share, Niranjan, would be is take pencil and paper and write down, lots of people have measured diffusion of polymeric objects and polymers like mRNA. And you can write down a simple framework of thinking about, is diffusion alone enough? And when you arrive at a paradox that you saw mathematically, that diffusion would not be enough for mRNA transport in a cytoplasm, say for a large cell or a small cell, you would have hit the crux of this problem. And so the only answer that I sort of have is, when I hear a question like this, I would go back to pencil and paper and really see from first principles, take an average size of an mRNA, calculate what would be the diffusion time in which, and you can run this even as a simulation. This has been beautifully measured in bacteria, where we can see that every second a protein can be everywhere in a cell, which is remarkable to think about in terms of numbers. So I think that's kind of how I would look at it. It's possible that there are active transport mechanisms. Frankly, most of the stuff, we are really on the tip of the iceberg when we think about physical principles in molecular and biological context. So you might be on the right track.
Yeah, so the next question is from Vedavalli and Ravina together. What they're trying to ask, like regarding this global warming. So does the global warming has now started to change the sinking and the rising pattern of these dinoflagellates? Since the
Yeah, I
density
think,
of
the reason we did this recent Alaska trip was really focused on acidity. There are two big things that in the ocean are changing from a plankton perspective. One is the context of acidity of the ocean is dramatically changing. It's related to CO2, because when it dissolves, it forms an acid. And so the pH of the ocean has changed significantly. The second thing that is, of course, is temperature. And the current experiments that we did, off the coast of Alaska, were all around modulating the pH in our machine and observing the sets of behavior to see what are the critical pHs around which these sets of phenomena change. And so I think, we know so little about how life on the microscopic scale works in the ocean, that even framing these questions, we are flying blind. It is very difficult to say any of the context of adaptation. It might be that the ocean has seen far worse and all the plankton life will be fine, or it might be that we might have a catastrophic ecosystem failure. We don't know, because we don't have the principles, we don't have the kinds of funds to actually study life in the ocean in the same way that we apply and in the same level of detail. So I think the fact that the best carbon sequestration technology on the planet today might actually be operating at its tipping point or has a lot of capacity, we don't know. And I think this is what drives me in this field, because we have to build better models. And again, it is not just the sets of global warming comes in a very complex ways. For example, literally 2 days ago, I read a paper. California is a very productive environment because many a times I talked about these nutrients are a little bit deep and light is on the top. There are these currents called upwelling currents that can bring the nutrients up from very deep waters. Suddenly, life blooms dramatically. And what we are just finding is that it's possible that these upwelling currents might be subdued, and suddenly zones in the ocean that look like rainforest can turn into deserts overnight. Just like when you think about ecology, you think about the formation of Sahara. There are Saharas in the ocean as well because there are no nutrients. These are called nutrient depleted zones, oxygen depleted zones. And overnight, you can have, as classes of, just the global circulations is starting to change. And ironically, we won't even see it because the only measure that we have on life on the ocean globally is just satellites, which is just chlorophyll in the top layer. You're just watching the skin, and we're completely missing out what is actually happening in the bulk of the ocean, which is, more than 90% of all inhabitable volume on our planet is underwater. So the terrestrial ecosystem, although very important, we give it a lot of importance because we live on it, might not be as important from a geochemistry perspective.
Yeah, so Manu, based on the answer that you gave, so I have a quick question regarding that. You are saying that we are only looking at the skin of the surface based on the color, like the green color. So are not can't we monitor this using spectroscopically technique where you're using various wavelengths, like certain wavelengths are being, I don't know whether it is going to work, whether like certain wavelengths.
Yeah, I think our current metrics is, there is hyperspectral imaging. There's all kinds of tricks that we are applying. And I think this is very important, combination of biology and satellites. When I think about programs in India on launching sets of satellites that should be really dedicated to biological questions, there does not exist a technique currently that you can measure the complexity of life in the ocean just from space. We need to be out there. And I think one thing that I am excited about is possibly with these new sets of marine biology programs that India is championing, there is budget for it. Hopefully we might have a fleet of dedicated research vessels. I mean, Indian Ocean is the most understudied ocean. Just it pains me to even sometimes think about it, is how much we understand about the Atlantic Ocean compared. Just we don't have enough people thinking about these questions. We don't have the kind of resources. There's no silver bullet to this. If you want to understand the ocean, we need people actually there. We need floating labs. We need new ways of getting many of you who think about molecular questions driven by human health to think about planet's health, because planet's health is actually deeply connected with human health. So, I think the idea is all flowers need to bloom. We need a massive growth in technologies to measure life in the ocean, applying all sets of principles that we have applied in optics. I think there's a lot of excitement. I don't see the kind of support as yet. That's the problem.
So there are few hand raised from the audience. So
yeah, it would be fun to hear from people.
Yes. So can allow the Raj Convert to ask.
Hello, can you hear me?
Yes, Raj, go ahead.
Yes, so I'm Jyotin actually. I'm attending from Raj system.
Yes.
Yeah, so very nice talk. Very interesting. Yeah. So my question is regarding this vertical migration of this Dinoflagellate. The hypothesis is that the nutrient availability is the depth dependent, right? So since we have these organisms in the lab, can we test it in the lab, in the pool which these organisms are stored? If we change the nutrient of that medium, then that behavior will emerge. We minimize the nitrogen content, then they will come down. Or
I think an absolutely brilliant question. We are exactly trying to do that. Unfortunately, the strain this is why we capture wild strains and lab strains. The strains that we have been isolated for 30 years ago. And ironically, behavior can be lost because they have been growing almost in nutrient-rich environments forever. And so I think we have isolated a strain that we are trying to set up directed evolution type of experiments in the lab, exactly as you described. Because I think, and ironically, we have some ideas of how to do it because, in the end, there are many parameters in the ocean, and 100 meter is just a challenge to think about. These are slow, long migrations. But and again, because cell cycle cannot be reprogrammed so easily, you end up thinking a little bit about what spatial length scale should you do that lab experiment at. And so I think initially we will do it temporally. And the gravity machine is designed to do that directly. So when I describe that loop, we actually have fluidic access to that loop. So we can in the wheel itself while we're observing. This is what I gave the analogy of an Oculus Rift, these virtual reality glasses. What I mean by that is we can change these parameters in the wheel itself.
Thank you.
So, hi Manu, there are a few questions still left. Few of them you're typing. So
yeah, I'll keep typing on the chat, but anybody else who has questions, I can take questions.
Okay. So there is one question from Casey, and what he or she is trying to ask, like rupturing of the wall, is it mechanical or enzymatic? If enzymatic,
yeah, I believe it's enzymatic. We showed mechanically rupture is equivalent just to show that the rupture of the cell wall is an important controller from a timing perspective. Why does it inflate at that time? The important aspect of that is actually made out of a remarkable material called dinosporin. We've been studying that material because we wanted to get things into the cell. It's very difficult to work with in the context of transfection microinjections, because you microinject, you destroy this thing, and then it doesn't heal. It's a cell wall. And just like you all know how working with plant cells is hard, and on the other hand, there's a very narrow window of time that we have where new dinosporin synthesis is happening, where we can get things into the cell. So we are trying that. But my belief is it's enzymatic. You can actually see it. In one of those phase contrast video, the outside shell was slowly disappearing. And of course, there is hydraulic pressure built in as well. So it's under stretch. I think, now we have the transcriptome. We are looking for, metalloenzymes, other sets of, if people have ideas, I would love to hear it. I think we don't have a candidate as yet. Because if we did have that candidate, then we can chemically show and demonstrate. If you can chew the wall from inside, I bet you can chew it from outside, which would be really fun.
So in the same question, like one more question is that, like in the post-rupture event, how a thin cell wall could be such a high pressure that is built from inside?
Yeah, I mean, dinosporin is a remarkable material. I mean, mechanically, it is just an incredible material. So in terms of just thinking a little bit about what's happening mechanically on this, it's just inflated, but it does not grow because it's just a mature polymer. The way you should think about this is that the inflation event only begins when dinosporin is enzymatically removed. So the pressure buildup won't be that high because this mechanical thing does not allow any influx as well. The calcium trigger is not there, the water influx is not there. So the pressure is not so high in the cell. When we pop them, they don't explode, they just kind of rupture away. The inflation happens post. I mean, it's beautifully aligned from a cell cycle perspective. You build up the pressure only because dinosporin was not there. And then when dinosporin matures, you actually stop it because then you've stopped the calcium and you've stopped that trigger in some sense. The extracellular trigger is stopped.
So Manu, while you guys were trying to rupture it mechanically, so the thing is only outside membrane is getting ruptured, or like once in a while you are seeing the entire cell was getting ruptured?
No, we don't rupture the cell because that would just damage the whole thing.
Yes.
Yeah.
So the thing is, if you're trying to do mechanically rupturing, so it means that both the cell layers, they are actually separate entity at that particular time of point.
During cell division, the cell rips itself off of the cell wall completely separates. And this is actually very visible in the data in the videos. You can see it separates out. It creates actually an extracellular space. We're trying to identify what fluid is in there, what is its density. But ironically, to access that, we need to rupture the cell wall. That's why we do these injection experiments. But that rupture event it was just a proof to show that the rupture is the trigger to initiate inflation.
So we have a few more questions. So Anuj is trying to ask, like, after Foldscope success, is there any new device especially for the masses in pipeline for future?
Yeah, there's a lot of work that we've been doing, and I think for the research community, there are two instruments that we built. One I was just showing was Planktoscope, and I'll just share this as I think if you all go to this website, we call this planktoscope.org. This is a new high throughput system for aquatic environments. We're very excited about building communities to map and measure water bodies. It's a high throughput system. It's completely open. Everything, instructions about building this instrument are available. It does require certain sets of electronic skills, but everything is documented. So if you go in here, hit replicate, you can assemble a kit yourself. Every little detail of this is provided online. So this is one. Another tool that we have just released that we will be describing in a lot more detail is Squid. This is, again, a way of thinking about bringing live and tracking microscopy to researchers, but very broadly, and especially reducing the cost, because every time you buy a microscope, you are stuck with that. And so this is another important event that you can look at. We will be announcing this shortly. It's called squidimaging.org. It's all the same sets of principles, but now higher capacity to be able to do a large number of experiments with a single framework. So, I think there's a lot of work in diagnostics that we are scaling right now that I didn't get a chance to talk about. I think we will soon have a new lab website with a much better documentation, because we realize sometimes just by publishing papers, people don't find out about these projects. So we have to document them very well. And then, of course, on Foldscope, the story is not done. We are only just beginning. There is roughly around 2 million users around the world, but there are 2 billion kids on this planet, 2 billion. So it's almost, we're a drop in the ocean, and quite literally. And, just purely thinking about India itself, if you look at the population of just UP, a state that I grew up in, it's roughly the population of Brazil. And you start realizing just we have a tremendous capacity in the context of just youth on our country. And it would be very valuable to make sure that we're thinking about, because it's not about the tool per se, it is about a community that's scientifically engaged. That's what we are really trying to do. It's about scientific literacy. If we believe that reading and writing and mathematics should be taught to everyone, I strongly feel we have to have a scientific aptitude and a way of exploring your curiosity, and a process of science should also be accessible to everyone. So often enough, people ask me what's next, and my answer is I'm still doing what I started doing 10 years ago. And I need all of you in being able to, I really meant it, what I said in the very end. The future of science in India depends on you. But remember, it's not going to be just the discoveries you make. Many of you will make remarkable discoveries, and that is phenomenal. But you have to look beyond that, because your biggest impact would be the kind of people that you mentor. And can you bring science to people that without your help and support would have never, ever engaged in the fold of science? It takes some gray white hair to realize this fact. But I can tell you already, in your future, if that is something that you actually do, the level of satisfaction that you have. And again, the irony is that if you do that, your science enriches in ways and forms that you don't even think about. Just take an example of try explaining your PhD to your grandfather or your grandmother, and you will immediately realize, what is it that you're trying to ask? Why is it so exciting? And can you really put that in the context of recreational biology? And you will learn something in that conversation that you might not have appreciated. But I feel as scientists, we don't do this enough. We complain about a lot of things, and it's all good that we should complain about. But the future of science in a country like India will depend on many of you thinking about, what is the circle of who I call a scientist, and can I change that? So, I think we're not done with full scope yet.
So actually, we are having lot of questions, interesting questions, and we still want to hear you. But
no, I'm happy to talk. I have some time. It's okay. Yeah, I think these conversations are rare. If people want, they can turn their videos on. This is informal. You can stop the YouTube streaming or something. I'm happy to talk for a little while. This is as much fun for me. I know it's late for you guys, but I have some time. It's okay.
So I'll just read a couple of questions. So the one question is from Siva, that he's trying to ask that while more nutrients are in the deep ocean, but the division starts and the density starts to increase near the sea surface level, how one can explain the nutrients needed to increase density?
No, I think the way you should think about this is that in the end, the cell is making this decision of how you would organize your cell cycle. We are looking for nitrogen storage mechanisms in this cell currently. So it doesn't mean that just because you are in a certain, and when I say nutrients, remember it's a photosynthetic cell. So it doesn't need that much other than the certain sets of, for all cells, and especially this is very true in the ocean, there are rare elements that you actually do need. And if they're not there, but then you can sequester and store them. So I think what we do believe is that deep dive does the storage. But then much of the growth can only happen when light is present and you can actually do photosynthesis. And that's really the dilemma, that you go a deep dive, you store what you want, you come back up, and then you spend 7 days and you say, oh, it's time, I'm out of things. You go back again. I mean, why do the division at the bottom is because only during this time of division, you don't have dinosporin and you have the capacity to inflate. You don't have any other time. You have a cell wall. You cannot use this idea of ballooning. And without ballooning, you can't come back up. I mean, think about it. Even if you are in seawater, you just don't have any mechanism of rising. And this is now a little bit speculative. We have not done the numbers properly, but we are trying to measure the amount of energy that it takes, because I mean, there is work done in inflation and there is work done in swimming, and we are trying to understand this type of a buoyancy engine. And I mean, any of you who, if you go study how submarines work, there's a lot of this going on in submarines of how they actually choose their position by taking in water, pumping water out, doing all kinds of funny things about. These are called buoyancy engines. We're trying to figure out this cellular buoyancy engine, how does this compare energetically to the swimming that we also study? But the amount of work done in rising against gravity, 100, 200 meters by a flagella, might have too much of a metabolic cost. As compared to here, there might be a different way, because you are really riding the wave. Well, there's no wave, but you are exploiting your environment in a very unique way. But it's not possible. The only time you can do ballooning is during division. So that makes sense that the cell cycle is positioned in that way.
Yeah, so we have